So it is my pleasure to receive it. It is our pleasure to receive you. So unfortunately, Sophia could not be here, but she sends her high regards. And in this case, we will have the pleasure of having then Joana, which has a PhD in theoretical and computational neuroscience. So we'll have a more biological-oriented talk, which I think it's really interesting. I even had, let me even say this to Joana, I even have a student of myself that saw your topic and wanted to ask to come inside the auditorium. Can I see it? Can I see it, please? So I trust that you have a very interested audience. Okay, so you are now working at the Life and Health Science Research Institute at the University of Minho, which is very close. And funnily enough, the speaker which is closer to us physically on daily time is the one talking online. So it's the fun of these times. So I will let Joana talk, and then in here I will try to be paying attention, and if someone wants to ask any question, I will use a microphone and make sure that you can listen, okay? So the floor is yours, Joana, and anything that you want from me or the organization, I'll be paying attention, okay?
Okay, thank you all. So first of all, I want to apologize. Yeah, it was a coincidence that this week, for coincidence, I have to be in Barcelona for another talk. I will start sharing my screen. Are you seeing it? Yes?
Yes, we can see it.
Yes. Okay. So I was asked to give this tutorial on network neuroscience, and I will briefly explain I will introduce you to what we understand as brain network dynamics, what are the brain network models that have been developed recently to try to understand the origin of the complex brain network dynamics that is observed, what are possible clinical applications, and some discussion. So first of all, this is the brain, right? And the brain is composed by over 100 billion neurons, the human brain. And these neurons are connected by like 100 trillion synapses. So this is arguably one of the most complex biological networks that we observe in nature. But then what is most striking is how this really complex network structure, also complemented by different types of cells, astrocytes, glia, neurotransmitters, and that. How does this translate into function, brain function in the sense of these nearly infinite activity patterns that coordinate behavior? So this is the main question that I've been trying to address over the last 15 years of research. So just to give you an overview, oh, sorry. So we have, sorry for ignore the bottom right plot. So we can have images of the brain structure. So these are like static images. We can have it from X-ray, where we see the bones, CT scans. We also use X-ray, but in higher resolution and more slices. Magnetic resonance imaging, usually this anatomical images allows to see the soft tissues. We can even do contrasts that are sensitive to angiography, so to the veins and the vessels. And also there is a technique called diffusion magnetic resonance imaging, where we can really see the fibers connecting brain areas. But all these are structural images. And if we think of activity, or what we understand as activity patterns, they can be detected by measuring signals over time. So we can use what is termed functional magnetic resonance imaging, which captures changes in the local magnetic susceptibility of the brain that is associated to the metabolic response of the brain. So more activity in some brain areas will give more signal, just in a very, very simple terms. Or we can also have measures of electrical or magnetic activity recorded using electrodes on the scalp. So these are the non-invasive tools that we have to detect brain activity from imaging, okay? So when we record brain activity, we can look only at the activity at a single brain area, but what is growing in the field of network neuroscience is to look at how different brain areas interact together. So usually what has been termed functional connectivity is when we look at the correlation matrix, let's say, between different brain areas. So for example, we parcellate the brain into a given number of brain areas. Here, for example, are 90 brain areas in this matrix, and then we compute the correlation matrix. But this correlation matrix, what has been shown is that it is dynamic. So one thing is to compute the correlation over the entire recording time, but we can also see that over time, some brain areas synchronize together and then desynchronize. So there is this expansion into the temporal domain of this dynamic functional connectivity that is really interesting, but also at the same time extremely complex to analyze. So it suggests and it indicates that there is a transient coupling between distinct subsystems, and this has a relevant function for the brain, as I will show in more detail. But this is just to give an overview of what we term of brain network dynamics, is this constant reconfiguration of patterns of synchronization between different areas in the brain, between the signal in different areas in the brain. And as you can see, it forms some network patterns that at the first look may appear random, but as you will see, there is some structure in it. So if we look from general perspective, what people have been analyzing in spontaneous activity, so this is without doing any task, just the brain, we see that there are some groups of brain areas that more consistently activate together over time. So for example, the visual areas will activate together. There's also the somatomotor network. So these are all networks, intrinsic networks in brain activity that have been detected. The dorsal attention, the ventral attention, the limbic that is more related to emotions, the frontoparietal is associated to executive control. The default mode is more associated to baseline activity. So it's mostly detected with stronger power when we are at rest, and then when we do a given task, the default mode goes down. It is still unclear the exact function of the default mode, but it's just to give an idea of when we talk about intrinsic functional networks or also resting state networks. But then one of the most triggering questions is, do these networks have a psychophysiological value? Like, do they have any function? And also, what drives the formation of these patterns? So these are the main questions of research in network neuroscience at this stage. So from analyzing fMRI brain activity, now I will go in detail how we define these networks and how we can characterize them. So I defined in 2017, while analyzing trying to develop methods to analyze brain network dynamics, we focused on the leading eigenvector at each instant of time. I will go in more detail later, but this is what allows us to capture these patterns of synchronization that are being formed over time. And this has allowed to detect, so if we apply a clustering algorithm on these patterns that occur at each instant of time, we find these recurrent patterns of activity, like the default mode network, the frontoparietal network, the limbic system, that consistently appear in different data sets across different subjects. So there are open source data sets from with fMRI activity from 400 people, healthy people, also people with psychiatric disorders. We've applied this method to over 30 data sets. There are some similarities between the different methods that exist, but most of all, what they allow is to extract from the complexity of brain activity that we detect the network dynamics that is underlying it. So we've used it already in some papers. So what does it mean when we talk about this leading eigenvector dynamics analysis? In a way, it's a method to characterize how the signal, the signal, when I say the fMRI signal, is the signal that we detect in the magnetic resonance scanner, how the phase of the signal, so if it's going up or if it's going down, aligns across the whole brain. So here, for example, each arrow, this is for one instant of time, each little arrow here represents how the signal is going up or down, but in the complex plane. So when we have a real signal, we can compute the analytic signals, so doing the Hilbert transform, and we get the phase of the signal. It's called here bold. So bold is also, I will use it as a same name given to the signals detected in fMRI, because it's associated to the blood oxygenation level dependent signal. But basically what we find is that when some brain areas increase their signal, there are others decreasing their signal. And this gives this complex patterns of activity. Here, for example, the main orientation. So we take the leading eigenvector of this, which is like a vector. It's clearly just an arrow that represents the main orientation of all the phases. And then we see which regions are shifted with respect to the direction of the leading eigenvector. And this is what determines these different colors. So in a way, if we list all the brain areas, so here we had 90 brain areas. So on the right panel, we see this leading eigenvector that is captured, and we see all the brain areas, and the ones in blue are the ones that are aligned to the main direction of the eigenvector. And the ones in red are the brain areas that are shifting from the main direction. So imagine if all the signals are going down, these are going up, or vice versa. So it is not necessarily a co-activation pattern, which is another method that exists, but this captures like whole brain patterns of phase coherence at each time point. Now, if we take these regions that are shifting their signal with respect to the rest of the brain, here I'm representing it on the cortical surface, we see the formation of a network. Here is the visual, is in the occipital lobe, let's say it's a visual dorsal attention. Now we take at each instant of time, we take these leading eigenvectors in each fMRI scan, and then we can visualize. This is exactly, so these bars is exactly what I'm visualizing in this video, just to give an intuition of what they represent. So the areas in red are these areas that are synchronizing together out of the rest of the brain. And we can, so here is a summary figure, and we can then cluster these. So at the bottom in panel C, we can cluster these signals, these vectors that we get using, for example, k-means clustering. It is working very well to cluster and to detect which are the centroids, which are the main patterns that reoccur across all the scans that exist in this sample. So if of course, when we do a clustering algorithm, we need to define how many clusters we need. And this is usually a problem because you can use optimization algorithms to define the optimal number of clusters. As I show here in panel C is the silhouette value that tells us, okay, with up to six clusters, we have the optimal silhouette value. So it would tell us the best K here. So if we have K equal 2 is the first line on the top, is we only want two networks to appear. And in this case, we see only one mode, one network is the global mode. Everything goes in lines, and we detect the default mode network as the second network, most probable. If we go with K equal 3, we detect the global, the default mode, and the visual network. With K equal 4, we start detecting more networks. So for example, with K equal 6, we already have some partitions of different subnetworks. So this is also to give an intuition that it is not a fixed number of networks that exists in the brain. And sometimes it's important to explore larger numbers of networks, so to be able to have more sensitivity to network patterns that are very specific to some disorder or to some condition. So these networks that are detected with LiDAR, they overlap with resting state networks. So this reinforces the validity of the method, because the resting state networks that have been described in the literature have been defined using different algorithms, using independent component analysis, using principal component analysis, using hidden Markov models. So that there are different approaches to operate, but irrespective of the method, there seem to be some patterns that consistently appear in brain activity. So we always have the first state. So here is called phase locking state, is when the whole brain, so the whole network system is synchronized together and everything is going up and down at the same time. Then another pattern that emerges is this default mode, when some areas in the frontal areas lock in phase, out of phase with the rest of the brain. So and this is to show the overlap with known resting state networks. So what this type of analysis, and that I want is the take-home message on this part, tells us is that brain activity can be approached as like the exploration of different attractor states, so different network configurations that recurrently appear over time across different subjects. So it's a system, a network system that is not static. It is not locked in a single network pattern, activity pattern, and it is constantly switching between different network configurations. So in a way, if you are more familiar with these trajectories into state space, imagine each state is represented by a network. So over time, we can characterize brain activity as a trajectory in a state space. So you are never necessarily falling into the centroid exactly into each pattern, but we approximate the default mode network, then we approximate the visual network. And this occurs at an impressive robustness and consistency across scans and across participants. So it's very interesting because we can characterize for each individual subject how it is exploring this state space. So just to give you here an idea of how the analysis is processed. So we have this recorded signals in these brain areas, and then we can find at each time point when a given network pattern occurs. So when we are closer to these network patterns. So here, for example, all the instances over time that are highlighted in blue would correspond to this type of network configuration, which means it's the occupancy of the limbic network. And then we can calculate how often this occurs. So the probability of occurring in each scan, and how long when it occurs, how long does this pattern last? And also, what is the probability of switching, of being in one network and switching to another? So it allows to make comparative analysis between conditions and disorders, for example, between patients and controls, between doing a task or no task. We can see if the expression of these functional networks changes across conditions. So some of the measures that we use, like I mentioned briefly, was that I said, okay, it's the probability of occurrence. This probability of occurrence in the language of dynamical systems theory, it's called the fractional occupancy. So how many time points from the total number of time points are assigned to a given state. Then we also have another measure called the dwell time, also comes from dynamical systems theory, and it's what is the number of consecutive time points that a given state is active or dominating in each session. So each scan, each session. So we can calculate these measures for each single scan, for each subject. Then we can also measure the trajectories in this state space. So in systems with multi-stability, the dynamics can be characterized using so-called Markov chains. Probably some of you have heard about it. So basically, it is the probability of being in a given state, switching to another state. So this is the first one, is the probability to switch, and then the Markov chain transition probability matrix is this value divided by the probability of being in the first state, in the original state. So this gives us a transition matrix that allows to try to interpret brain activity. So for example, using this is the probability of being in state 1 and remaining in state 1. So you have 77%, and this is represented on the right side as a graph, illustratory graph that represents the matrix on the right. So once we are in the global mode, most probability in the next time point, the signals will be in the global mode. Then with like 8% probability, we see the transition to the default mode network. And then there's around 50% probability that it will remain, or it will descend back to the global state. And for all the networks that appear that are not the global network, we find that there's always less probability of going to those states, but then when they reach that state, there's always more probability to return back to baseline. So this has led to the idea that the baseline state, this global mode of activity, imagine any network, it can be the brain, but any network that has like a globally synchronized mode of activity. And then from time to time, it switches to patterns that are like locked in phase and it's almost like a vibration mode in a network. And these are termed, we term them ghost attractors. So it's one term, or pseudo attractors. It means that these patterns emerge recurrently, consistently, but only very briefly and erratically. So they are there is no doubt. They overlap with known functional subsystems, but why they appear and their emergence is still unclear. But these insights from using dynamical systems theory to characterize brain network dynamics is important in the sense that it gives us like a framework to try to understand the stream of consciousness in the brain. And there have been more and more works trying to interpret brain activity as this trajectory into state space of brain network dynamics. So here I'll give you an example of what we do when we compare, for example, the probability or fractional occupancy, depends on the name that we use, of the occurrence of these network patterns. So this was with a data set where subjects were given psilocybin. Psilocybin is the active ingredient in magic mushrooms. So this is a collaboration with the Center for Psychedelics Research at the Imperial College, where they do these analyses. But it's important because these pharmacs or these psychoactive compounds have a really, really strong effect and can be given in very controlled experiments to healthy humans. Of course, with all the ethics, it's not, I don't do that part. I just get the data. But what I want to show here is very important. So before and after placebo, the little bars that we see are the error bars. So it's the mean and the standard error of the mean in all the subjects and all the scans that were done before a placebo was injected and after a placebo was injected. So there is no significant difference between the pre and post bars, as you see, like the red bars are mostly identical. Now, before the injection of psilocybin, we also didn't detect any significant difference. So there's no asterisk anywhere. And this was done 2 weeks apart from the placebo session. So it's interesting that there is a quite strong stability, or we call reliability, of the probability of occurrence of these networks across sessions. Now, when the subjects are inside the scanner injected with psilocybin, we see a strong change in the expression of only some networks. So the global mode increases, but specifically what we find is that there is a almost disappearance or a significant reduction of the third, here the third phase locking state, which is the frontoparietal network. So it's a network that links the lateral frontal with the lateral parietal areas of the brain. And this network significantly decreased in mostly all the scans that were done after psilocybin. And when this network disappeared, what we see is more of the baseline. So it also aligns with these theories that we have these excursions to these patterns, and the common baseline is the state one. So now I will explain, then before going into the clinical explications, I will explain about these brain network models and why they are relevant to try to understand what we are looking at. So far I talked about this functional connectivity, connections between dynamic signals, right? So it is not static, but what supports and what gives rise to these dynamics? So first we need to understand what is the underlying network structure, right? So we know there are fibers connecting different brain areas, long range fibers, and these are thought to play a very important role in not only in shaping the correlation structure, like in terms of static correlation between different brain areas, but also in shaping its dynamics. But to what extent does this network structure shape the dynamics of the brain? So this is what I've been doing, following, of course, the seminal works by Victor Girsa, Gustavo Deco, Michael Breakspear. There are some groups, so this has started maybe in 2008, are the first brain network models who try to understand the origin of these macroscopic network patterns. So we use these reduced network models. It's just to give an overview. So in a way, the structural connectivity, when I will talk about structural connectivity, is the number of fiber tracts or fibers, right, that are detected between each pair of areas N and P. And the functional connectivity, in neuroscience it's called like that, is the correlation between two brain areas. This functional connectivity can be dynamic, we can use sliding windows or use this instantaneous phase coherence. But overall, people, until maybe the last 5 years, 10 years, we're just looking at the static correlation, and termed functional connectivity is this correlation between different brain areas. But what is the link between structural and functional connectivity? And this reports to the big issue in neuroscience and even in physics. It's the structure-function relationship. How does structure give rise to function, right? How does cognition emerge from the physical structure of the brain? So it's not just about finding a mathematical transformation rule to go from one to the other. Because at the beginning, there was some people trying just to see how the structural connectivity could explain at the static level, very immediate. The thing is that we need dynamical models. And this illustrates this in the sense that this is a complex network dynamics. And as you know, it's an expanding research field in physics and mathematics. And there are still many puzzling phenomena that remain unexplained. But importantly, over the last decades, there have been more and more projects and works trying to understand the dynamics of networks, right, of complex networks, and how the synchronization reveals these topological scales. I don't know if you've heard about metastable chimera states, but it's, for example, when a group of brain areas, for example, here the yellow are nodes, a group of nodes, because this extends beyond neuroscience, when a group of nodes synchronizes together and then desynchronizes, and then another group of nodes synchronizes together and desynchronizes, well, whereas the whole network is never fully synchronized. And there are very many interesting works that serve as a basis for the interpretation of the results found in neuroscience. So but one of the main things of this network dynamics is that the network structure alone cannot explain network dynamics. We need dynamical models. And what are these dynamical models? There are three major ingredients that play a role in shaping network system dynamics. So it's the structural connectivity, right? So as you know, like the adjacency matrix, but here they are weighted, the networks. The node intrinsic dynamics, which is each node represents a dynamical system composed of millions of interacting neurons, and it has an intrinsic dynamics. And it's important to know how this behaves in the spontaneous state. So if these nodes were uncoupled, how do they behave? And there are different reduction lines in the sense to try to describe with a mathematical equation. So we use differential equations to explain to describe the behavior of each node, right? So there are some models like the Wilson-Cowan model. I will go, I think, into detail on this, but it's how the different excitatory neurons influence the inhibitory neurons, and then this creates a feedback inhibition that generates an oscillation. Or there are more reduced models even that consider directly oscillatory models. So it's this Andronov-Hopf models of metastable damped oscillations or sustained limit cycles like the Kuramoto model. And then, so in terms of intrinsic dynamics, yes, so here it's important to take into account that each node in this network, in the brain network, already represents millions, if not a billion neurons together. And they can be seen already as a local dynamical system. And this local dynamical system exhibits collective behavior, can exhibit collective behavior, like the emergence of slow with respect to the fast spiking of neurons, oscillations in the local field potential. Usually the main oscillations that are detected at the single node level, so with an isolated group of neurons, so you can generate it even in vitro without the brain around, is the emergence of oscillations at 40 Hertz. It has been proposed as being like the natural frequency of neuronal masses, but it is well explained now due to this feedback inhibition between neurons. So the idea is that we can assume that brain areas will engage at the local level, each node have the ability to engage spontaneously at 40 Hertz. Now, how do these nodes interact together? So this is also the model from Wilson and Cowan, how these networks, these oscillations emerge because of the coupled excitatory and inhibitory systems. But how do these systems then interact together at the global network level, right? How does the input received from a coupled node is integrated? And this is where we need to generate these equations. So I will explain how to generate the equations that describe a whole brain network model. So in a way, it's a system of coupled differential equations where there will be terms describing the intrinsic dynamics of each neural mass. So those are the nodes. There are terms describing how the input received from other neural masses is integrated. And then depending on the model, we can take into account conduction delays, noise, even the concentration of neurotransmitters available. So there are many ingredients that then can be added subsequently to the network models. But maybe the most simplistic approach, and I will show a bit more complicated methods, is to just look at the brain as a system of coupled oscillators. So if each brain area has the ability to oscillate at 40 Hertz, what happens if we couple these 40 Hertz oscillators in the network structure? So we can reduce the dynamics to a simple phase oscillator. And by reducing to a simple phase oscillator is that we can use the Kuramoto model of coupled phase oscillators. So it has, despite its simplicity, so the Kuramoto model is considers that each node in a system depends on the phase of the nodes to which it is connected. It can give rise to broad range of phenomena of dynamical results that are very interesting. And we can simulate and reproduce really complex phenomena that are observed in nature. There have been many complex behaviors reported from synthetic studies. You know, you just change the connectivity structure, you have different networks with different modularity. You probably even know better than me about network science and how to couple these different systems. And this is useful for neuroscience because they have been solved analytically. Not all, but some of them have been, and this can help us interpret the results. So, and then the coupling function is still also kind of unclear, but what is known is that there is a long-range transmission between signals. So this transmission is not instantaneous. It's important to take into account as well the transmission delay. And this is very important, as you probably know, because if you have a network and there is no delays between any nodes, everything will only synchronize together. But once you have delays, it will create these feedback loops that then generate really complex dynamics. So we take into account not only the coupling weights between brain areas, so we have a matrix with coupling strengths that depend on the number of fibers detected, but also the distance or the mean fiber length between each pair of areas. And with this, we adapted, so I did this in 2011 during my PhD, was to adapt the Kuramoto model to the connectivity structure of the brain. So basically, we have a natural frequency for each node, n, which is 40 Hertz, for example. We can put it all identical, or we can choose a distribution of frequencies. There are different variations that can be done on top of this. Then there is the interaction term. So if K, like this global coupling here, this global coupling strength was 0, all we have, each oscillator is just oscillating at its natural frequency and it's unchanged. Once K is above 0, each node starts receiving inputs or being influenced by the activity in its neighbors. So here it's the connectivity strength between each pair of areas N and P, right? So all the nodes that are connected to area N, we will take into account. And then we take the difference of the phases, but the phases with a transmission delay. So the original Kuramoto model didn't take into account delays, but more and more there are works showing the importance of these transmission delays in the dynamics of a system to frustrate full synchrony, but also to generate interesting activity patterns, as we will see. And so this is basically how the equation works. And then we simulate this and analyze the results in the same way as we analyze brain activity. So they are here just to give an overview of different brain network models, where they all can be resumed to the variation of the signal X in each node N. So on the top here, the big equation, over time, right? So over time, at each instant of time, takes into account a neural mass model, you can choose from more simple to more detailed, plus the input from coupled neural masses. So there is the Fitch-Hugh-Nagumo model, the dynamic mean field model. It takes into account the concentrations of neurotransmitters such as NMDA, the Wilson-Cohen model is just a firing of the different excitatory and inhibitory neurons, Hopf bifurcation model, which basically is it's called the Stewart-Landau equation that describes the emergence of oscillations, but whereas in the Kuramoto model, the oscillations are always self-sustained. So it's always like just an oscillation and there is no information on the amplitude. The Hopf model takes into account the amplitude fluctuations as well. So we are now working also mostly with models that take into account the amplitude of the oscillations. But the Kuramoto model gave a really nice insight. And as you can see in all these models, there is this coupling term. In some equations, the global coupling is called K, in others is called G, but the dynamics of the network depends a lot on this global coupling parameter. So the coupling that scales the interactions between units. If K is if the global coupling is very close to 0, there is little integration, in the system. So the nodes don't interact together. If K is very strong, everything is synchronized together. And it's in between, there's a critical range of coupling between brain areas that generate the really complex behavior very similar to what we observe in brain activity. So this has been linked to these theories of segregation and integration. This is what I illustrate here very briefly, but when you have lower coupling strength, you have mostly connections between nearest neighbors. As you increase the coupling strength, you see more and more the interaction between distant groups of nodes, and with strong coupling, everything is synchronized together, everything is coupled together. So here again, it's a way to analyze this, where we see, for example, the level of synchrony. I don't know if you're familiar with the order parameters. So we look at the signal in each node, and we see how aligned the phases are. If everything is aligned together, they are synchronized. It's here represented by the magnitude in panel C of this line, this black line. So for K equal 20, you will see that this magnitude of the order parameter is larger because most of the phases or the signals are aligned and they keep rotating. So when it's represented like this in the circle, it means that if we would do a video of this would be rotating, but always with a large magnitude. And for weak coupling, all the phases are distributed and there is no synchrony at all. So here, as they show, there is this increase in synchronization level as we increase the coupling strength. But more importantly, there is a measure that has been used more and more in neuroscience, in network neuroscience, which is called metastability, which is in between incoherence, where nothing is synchronized, and full synchronization. There is this formation of clusters, so metastable cluster synchronization that's kind of synchronized together, but then desynchronized. And then this other synchronized together and then desynchronize. Also aligned with the ideas of chimera states that I talked earlier. And it has been suggested more and more, so it is believed that the brain network supports this metastable cluster synchronization. And what we are observing in resting state activity with fMRI is the signature of this metastable cluster synchronization that is occurring in this range of parameters when humans are awake, because then When you are fully anesthetized or with loss of consciousness, this changes these parameters. So again, this is another illustration of what means the incoherence, the metastable synchronization, or for high synchrony, everything is synchronized. So this has led to the development of new theories about brain function, that not only the brain seems to exhibit multi-stability, so there are multiple patterns that can be detected. It is not stuck in just one pattern. And so there is this observation of multiple recurrent functional connectivity patterns or functional networks, and also metastability. Metastability in the sense that when you activate a given these networks, these ghost networks, they don't last very long. It's not really a stable attractor. It doesn't stay there. It lasts. It is short-lived. Now it is still under debate if this short life is due to noise. So in this, even from quantum mechanics, here we have the dwells of attraction, and then sometimes to jump from one dwell to the other dwell of attraction, you need noise. Or sometimes it's really the intrinsic stabilities of the system, because it's such a complex network that it will never stabilize at a given configuration due to these feedback interactions and noise and heterogeneous connectivity, that it is constantly switching between different patterns. So this is the ongoing, I would say, hot topic in computational neuroscience. Here it's hard to put together all the models that exist, but in 2017, I wrote a review about the existing brain network models. And so here they are ranked from top to bottom by the level of complexity. So reducing the complexity from top to bottom. So at the top, we have models of spiking neurons. And really a model of spiking neuron, you take into account maybe in each node, you can put like 1,000, 10,000 spiking neurons, 1,000,000 spiking neurons, right? But without any reduction, you really simulate. So it has high computational cost, but it considers each neuron is an integrate and fire equation. Integrate and fire means that when the when the input is sufficiently strong, it will drive a spike. It will fire the node, the neuron. So it's non-linear, right? So the more input you reach, when the input reaches a given threshold, the node will fire and then it returns to baseline. There's a refractory period, all of that. You can take into account the neurotransmitters, AMPA and MDA receptors and all of that. And in this case, usually if the units are uncoupled, so a spiking neuron, if it doesn't receive any input, it will be just in a fixed point, in a static point. And then when they are coupled together, we see this multi-stability between fixed points. So this was initially the model of spiking neurons at a large scale was introduced by Izikevich and Edelman in 2008. Now starting my PhD, then Deco and Girsa wrote this paper showing the multi-stability and ghost attractors using these very detailed models. But then the idea was to consistently try to reduce the problem, to see what is giving rise to these macroscopic phenomena, and can we use more like neural field models where you just use equations to represent the whole field activity, and also these Kuramoto oscillators, or even just simply linear stochastic models and that. So the idea is to see which of these models, what are the theories that they give for the emergence of these slow fluctuations in brain activity that represent the resting state networks. So it's the theories, they are not exclusive. They can be all, they can coexist. So there are different expressions, but for example, just to say, the origin of slow fluctuations have been suggested to relate to the brink of multi-stability, intermittent self-organization from chaos, connectome-specific harmonic modes, so like vibration modes of the network itself, being in the vicinity of a supercritical bifurcation, and then when you have noise, the system engages into a resonant mode and returns back to baseline, metastable chimera synchronization, what I said about the clusters that synchronize, and overall this is it. Then there were some models, like here a linear stochastic model that was proposed, that just suggests us there are noise deviations around a fixed equilibrium point. So you always have just a fixed point. And in this case, it is failing to, because here I add a column about model validation and how much they are able to explain the different, the origin of the signals. And indeed, the more simplistic model, where it's just, for example, the spatial autoregression, is simply to take an equation that transforms the structural connectivity to the functional connectivity, adding a bit of noise. And in this case, it fails to show all the features that brain activity empirically shows us. So we have this puzzling empirical evidence that we need to make sense of. And these are an overview of the different models that have been proposed. But so despite the difference between models, there is a general agreement that the anatomical role, network plays a role, no doubt, of course. But it's not just the anatomical network. Also that the system is neither fully incoherent, neither fully synchronized. So it's a network that is somewhere in between. It's called this critical dynamics. So it's below, between these two critical points above which everything synchronizes or below which everything is asynchronous. And both metastability and multistability appear to be key factors of brain network dynamics. But there are many aspects that are still under debate, like the role of time delays, the role of noise, the node dynamical regime. What is the best one depends also on the question being addressed, the coupling function, if the coupling is linear, or if it's coupled by the phase, or if it's in the complex domain. All these different issues are an ongoing field of research in brain network dynamics. But then, all these networks that we model, they need to be validated, right? So we want to compare with brain activity. So we simulate the activity in computers using different softwares from MATLAB, Python, or anything. We simulate the activity, and then we treat the simulated signals as if they were the signals detected from brain activity. So we can use the balloon model is an equation that kind of smooths the simulated signals to replicate the signals that we detect with functional magnetic resonance imaging, which has this blood oxygenation level dependent signals that are intrinsically slow, but that reveal these macroscopic network patterns. Or we can band pass filter the signals that are simulated into different frequency bands to compare with what is seen with electroencephalography and magnetoencephalography. So this is just to show, so this is the starting point, the structural to functional, but beyond this, so we have this fit to the static functional connectivity, then the functional connectivity dynamics, which is how over time the phase synchronization matrices or the correlation matrices evolve over time. So temporal similarities of the signal, how much at each instant of time the signal is correlated to the past. Okay, so, and it's interesting because you see increases and decreases. There's usually this checkerboard effect indicating that there is recurrence in the dynamics. Memory, and also the spectral properties of the signal, which is the functional connectivity and the correlations between brain areas or interactions between brain areas also depend on the frequency band to further complicate the story. So, and these frequency bands emerge from interaction between different brain areas due to time delays. So one way to validate, for example, how we simulate these things is we compute the correlation between empirical data. So we have the structural connectivity, here C, we have the functional connectivity, and then we see as we increase the coupling strength, when do we have the best fit? So if the coupling is very weak, the correlation matrix is just random. If it's very strong, it is all red, means everything is synchronized together, correlated together. And in this intermediate coupling, we see this is where we see the structure coming out, the structural connectivity. Right? And then to compare the dynamics that is now called FCDs, functional connectivity dynamics. So one thing is to fit the static functional connectivity, the other is to fit the functional connectivity dynamics. And it has been shown that it allows to a better prediction of the fitting to find the optimal working point. This is also still under investigation, how to optimize these measures. But one of the things that I think at least I learned the most when simulating these interactions between nodes with time delays, is that when units, and this is important, like this is the take-home message, when units that oscillate at a fast frequency, for example, at 40 Hertz, but this can be different, when they are coupled with time delays and they synchronize together, they synchronize at a reduced collective frequency. So the frequency of synchronization is different from the frequency of the units when they are isolated. So for example, if all were at 40 Hertz, whenever the synchrony degree increases, so this order parameter is the synchrony degree over time, and here it's low pass, so that in red, so that we can see it very clearly here. So we see that, so this is time in seconds, sorry for this, time in seconds. So sometimes for 20 seconds, the synchrony degree is lower, but then the synchrony degree increases. And when the synchrony degree increases, we see here at the bottom, we see the emergence of peak frequencies that are at slower frequency, so at 16 Hertz. So this could explain why there are frequencies or oscillations emerging from the network dynamics itself. So from the structure, so it's the structure that is inducing these oscillations. So it's not a pacemaker structure. It is not a pulsation. It is really the network itself that when it tends to synchronize generates these slower oscillations. So this weakly stable cluster synchronization at reduced collective frequencies is now accepted as a mechanistic theory for the EEG and MEG brain rhythms, which was really interesting. I think it's the most relevant work I've had. So then in terms of clinical applications, I now want to, so going from the brain network dynamics, then these models, how can this give us insights into these different disorders? So one of the things that had been reported using graph theory, so for, I'd say maybe not so recently anymore, but the earlier decades, there was a lot of work applying graph theory to brain networks, right? But nowadays it's more moving towards the dynamics. But applying graph theory just to functional connectivity matrices, it was found that, for example, in people with schizophrenia, there were properties from graph theory of the network of the brain of these people that were different from controls. So patients with schizophrenia exhibit reduced small worldness, as you know, right? It's then the increased hierarchy, lower clustering coefficient, improved efficiency, greater robustness. Robustness is the robustness to attacks is when you remove a node, what is the size of the largest connected component, right? And they found that schizophrenia patients had greater robustness. So it was even suggested that it could be an advantage for of these signals, these patients. So an evolutionary advantage. We never know if these psychiatric disorders have only bad effects, but can have positive effects. But overall, when we read this paper has had a lot of impact. When I read it, it was like, why this is happening? Can we explain this in some way? And the thing is that the word schizophrenia comes from a splitted mind. So they have a lack of ability to integrate, right? The thought processes. So it's called really a disconnection syndrome. It is what is believed. So then came the question, can we reduce, if we reduce the coupling strength in the model, in the network model, could we observe the same graph theory changes that are observed in people with schizophrenia? And so that was the main idea of this paper. So how functional graph alterations in schizophrenia can result from this global anatomic decoupling. And then I extended this to model how if we remove different links and different things. But basically here I show the different measures from graph theory where efficiency is the inverse of the path length, right? Small worldness is the clustering coefficient of the network with respect to the clustering coefficient of a random network. Well, so I used exactly the same algorithms that they used in the Linal paper, in the clinical paper, to the model, applied to the model equations. And what we find is that, imagine this is the coupling strength, and the green value, that the green line here corresponds to the level of small worldness reported the mean in healthy people. And in red is the degree of small worldness reported in people with schizophrenia. And here in the model, we see that this can be explained by a reduction in the coupling strength. You see that it goes, we have values that approximate healthy values for a stronger coupling. As we decrease the coupling between units, we find values that approximate people with schizophrenia. And this was consistent for all the graph theoretical values that were reported in the clinical work. So in a way, it explains that what was reported as functional graph alterations in terms of graph theoretical measures can be accounted for by a single global parameter in the model, so a decrease in the global coupling strength. So it shows how the models can go hand in hand to explain phenomena that are observed with measures from graph theory that lack the mechanistic value, right? So it's a metric that is used to characterize systems, but then to understand where it comes from, it is important to take into account the models. And this model, this result was found to extend not just using the Kuramoto model, so decreasing the coupling, even using a more simple, not simple, but a more biologically plausible model where you use fluctuations in the firing rate of neurons without even time delays. We found exactly the same thing, so that reducing the global coupling would go from the values from healthy controls to schizophrenia. And not only reducing the global coupling, but also pruning. Pruning means like cutting randomly different connections of the brain. If we prune the network, different connections, also the graph theoretical measures that are detected could be approximate what is found in schizophrenia. So then there are two different scenarios here. Do people with schizophrenia is just a reduced global coupling, or do they have some connections that have been too much pruned? And this is interesting because it links to psychiatry. For example, a lot of schizophrenia episodes appear right after adolescence. And adolescence is characterized by a period when our brain, the structural network, is pruned. So when you are born, let's assume you could also, okay, in a very simplistic way, imagine that everything is all-to-all connected. So all the neurons are connected to each other. Then some connections become stronger as you use them more and more. And then around adolescence, the ones that are weakly used are pruned. So there are consistent reports showing that there is a decrease or a more structured organization of the structural network after a given period, the period of adolescence. And this suggests maybe the people who are suffering from episodes of schizophrenia at that stage can be related to the wrong pruning, but then schizophrenia appears in other conditions and in other periods of life. And in that case, it could be reduced to lower, associated to lower coupling or to other disorders. And then these models, so the model that I explained before was just considering connections, right? Without taking into account the modulation from external factors. So if you are also interested in networks and network dynamics, control theory is nowadays how you can control the dynamics of a network. And in the brain, it seems that what controls is this neuromodulation of the brain can be modulated by psychoactive compounds. Psychoactive not just in the sense of psychedelics, but also in the sense of psychoactive drugs for psychiatric disorders and all of that. So there are pills, let's say molecules that you can inject, you can inject, but you can also take just a pill. It passes through your brain and it will modulate the way your brain network is operating, right? So one of the ideas here was, again, in collaboration with the group from the psychedelics research, because these psychedelics have really strong effects. So it's easier to study. It's almost visual. We see the brain activity completely different. So in this case, it was healthy people taking LSD, which is it's an acid, let's say, on the brain. And then this LSD is known to bind to serotonin receptors. So there are parts in your brain, which is the map that you can see on the top right-hand side, that shows in which parts of this network LSD will bind with stronger strength, right? So it will go to the areas with more concentrations of receptors of serotonin. And only some type of serotonin receptors. It's a specific one, which is the 5-HT2A. So we have the real signal, we compute the functional connectivity dynamics, we can look at the distribution of values in the functional connectivity dynamics, and then we run the model and we compare the functional connectivity dynamics in the model with the one empirical. And what was found is that we have the best fit. So we first take the best fit to the placebo condition, right? So to see where we got the best fit of the FCD, and here it's to minimize. The best fit is when we minimize this value. And we found that if we don't consider the serotonin receptor maps, we fit well the placebo condition. Now, if we we increase the excitability, so it's the gain function, the excitability of the brain areas that have more concentration of serotonin in the brain, in the model, then we have a better fit. So this FCD KS distance into LSD works much better than if, of course, if we reshuffle the receptor map. We tested with different approaches, even with receptor maps from different neurotransmitters. And we find that this receptor map helps explain the response of the network of the brain under LSD. But this also is still ongoing research. These results have statistical significance, but they still are far from being really predictive, so to be translated into the clinics or the design of better drugs. But still, it's a promising area of research. So it is comparing with different receptor maps from, like within serotonin, there are different binding structures. But anyway, the prediction of the model was better when we took the right receptor maps. So this is summarizing this idea of using these receptor maps to design better therapeutic drugs. So if we have the whole brain model of a diseased brain, and then we can design what would be the optimal distribution of receptors in order to generate or to recover a more balanced brain network dynamics as associated to the brain. So in terms of discussion here, I would like to first say that despite this effort of doing computational models, there is a sentence that I think, okay, all models are wrong. And before people start, criticizing that these models are not perfect, but they are useful in the sense that they can help us not only understand the generative mechanisms of what we are seeing, and this, I term them phenomenological models. And I think the models of the brain network dynamics are still at this level, the level of phenomenology. So we just need a qualitative validation. We don't have an exact fit. But then the idea is the more we understand these phenomenologies that come from different components and different mechanisms that may coexist at the network level in the brain, they can be optimized over time and turned into predictive models. This is the goal. This is the main goal of network neuroscience and brain network models, is to help and simulate the outcome of therapeutic interventions in order to optimize, to design the stimulation strategies. CBT is cognitive behavioral therapy, but try to simulate all these things in order to have a quantitative validation. But these models can only be translated into clinics once they turn into good predictive models. So we are still far, not so far, I believe, I hope, but I see this at least as a vision for the future. I think there will be personalized, like in silico brain models, to help to predict the therapeutic strategies, or to at least predict the outcome for assisted decision in psychiatry, so that the subjects can enter an MRI when they have a psychiatric disorder, they can be scanned, and then using these models, we can try knowing what is supposed to be the healthy network dynamics, how we can tune what would be the best simulation strategy or the control of this network in order to recover and to rebalance brain dynamics to a healthy brain. So this is my overall, I hope this is so many things to say in 1 hour and a half, but I think I gave an overview of what is my current research at this level in studying and trying to understand brain network dynamics. And I would be happy to hear your questions and discuss further these issues. Thank you.
So thank you so much, Joana. So before we start our round of questions, we also have an online question that perhaps you might start with it. I can read it to you if you want, but if you press the questions and answers on your Zoom interface,
yes,
you should be able to see it.
Yes.
Potentially.
Do you want me to
read it? Okay. If you
or you read it?
But perhaps you should read it because it might be hard to.
I'm curious which dynamical model, Kuramoto, etc., you took for the metastable chimera states. Okay, it was Kuramoto. I'm asking because we find similar decrease in frequency of synchronized Rössler systems. Yes, chaotic dynamical systems. I think this might be indicative of a universal phenomena in synchronized oscillations. This is a beautiful question. I've exactly tried. So first, the results were found using the Kuramoto model of coupled oscillators, this reduction of the synchronization frequency of the collective frequency upon synchronization. And I was trying to understand because we could see this in the simulations, but I wasn't sure where this was coming from. And then I found there's a paper from Ernest Niebuhr, 1991. It's called metastability and collective frequencies in networks of limit cycle oscillators with time delays. And there, again, they showed this happens with limit cycle oscillators, like the Kuramoto model. But in the brain, it is known that brain areas are not just limit cycle oscillators. So I agree that we need more complex models like the Rössler systems or the Hopf model. And so I validated, so I was comparing the results doing with the Kuramoto with the Hopf model, but it would be interesting to do also with the Rössler to see if it's the same phenomena that is occurring. But basically, if you just correlate the peak frequency with the level of synchronization over time, usually we find a quite good agreement in this. It's highly significant. You can try to predict the synchronization frequency as well. There is a way to do the analytic prediction based on the Niebuhr paper. I actually just put it on archive paper that I'm writing on that. So if you want to look at on my Google Scholar or something, it's maybe the latest paper that was put on archive. It's called Synchronization in the Connectome. I hope this answers the question.
Thank you, Joana. So we have space now for questions from the audience.
Hello, thank you for the talk and sorry for my lack of eloquence. First question, I have a two-part question. The first is, when you dealt with patients with schizophrenia, was it the first onset or they were already medicated? And also the second part is, did you take into account, as you did with the LSD patients, LSD, people who took LSD, and reduce the parts that are most receptive to dopamine as to try to get the same results with patients with schizophrenia? Did I make myself clear?
It would be, I think so. So if the changes observed in schizophrenia could be associated also to changes in the receptor density of the different neurotransmitters. So first, regarding the first question on the subjects, if they were, so I used already, I work mostly with collaborations. I didn't do directly any experiments. I did only recently on rats, and still it was someone else doing it for me. So I do mostly the analysis part. and even now we are submitting a paper on schizophrenia where we take data from open source databases. So these databases are online. For example, there is one called the COBRE, where there is data brain activity from 75 subjects with schizophrenia. Sometimes it's first episode unmedicated, but in this case they weren't. But it's an important question. But it's very hard to have someone who is completely unmedicated, the so-called drug-naive patients. We have ongoing studies trying to collect a sufficiently good sample at the University of Minho, for example, of drug-naive patients with depression or obsessive-compulsive disorder. But it's true that medication affects the results. Okay, but in general, there is a feature in these patients. So even the drugs are not necessarily curing all the essential changes in brain activity. It would be interesting indeed. I don't know if there's a study already recently exploring the relationship of the receptor maps, density maps are done with a different imaging technique called PET, positron emission tomography, and I don't know if there's a study already looking at differences in the concentration of receptors in schizophrenia, and if this could explain the changes found in brain activity. So, but it could be an interesting field of research. Yes.
Thank you very much.
Hello, thank you for this great talk. Could you comment more on how can we use these dynamical systems, for instance, to have more theoretical and analytical insights in neurodynamics? For instance, can we use chimera states or model for chimera states to understand better the recurrent patterns that you showed in the first part of your talk?
Again, also very interesting question. It's true, like that. I mean, what I've been trying to do is to try to read the literature on these chimera states. I established like a collaboration with the Mathematical Institute in Oxford, where every time I have these signals from brain activity and we want to think of new ways to analyze them and to validate also the methods, because sometimes we think, what if is this a chimera state? What defines a chimera state? What defines a ghost attractor? You know, like it was also, can this be called a ghost attractor? And for example, I entered in contact with a professor of dynamical systems theory who was in Nice, Bruno Cessac, because I met him at a conference, and then I came there and we discussed, and he gave me the notes from this to calculate these dwell times, Markov chains, to try to translate these methods, these tools that already exist, right? We don't need to reinvent the methods for neuroscience. And so it's a bit a work of search and trying to bridge fields and try to better understand. Even the chimera, because now that you mentioned that, it has been like a hot topic of discussion, because, for example, I'm now in Barcelona with Gustavo Deco, and he was saying, no, chimera, you really need symmetry in the brain. And then we were reading, and indeed, until the 90s something, chimera states had a very specific meaning. But then it has expanded, and there are papers from 2006. So the term has gained a broader meaning, and it is sometimes very subtle to try to find if what we are detecting in brain activity are signatures of that, and how to prove it is. So at this stage, what at least my way of doing this is to propose that it may be related to that, so we try to make models that approximate this type of behavior, but then to make like the claim, it's very hard. You know, in science it's like this: you have a mechanistic theory and it remains valid until refuted, but it's hard to really prove a thing. So there are different beliefs, there are different possibilities to explain the emergence of these patterns. I am inclined to believe they are associated to at least complex network dynamics in general, or critical network dynamics. if it's specific to chimeras, if, you know, it depends also on the word itself, and these mathematical terms sometimes are hard to define. There are suggestions it's also related to stochastic resonance. I don't know, so with noise, you resonate in the network, but maybe chimera states are also associated to stochastic resonance, and they are also associated to the harmonic modes of the structure. So yeah, I think we need more people in the field and more people trying to approach these questions. So I hope it will come in the future.
So I'll ask a question myself, Joana. So as a first side note, I want to say that incidentally, I started work on network science precisely because of brain networks. When I was a PhD student, I was sent to Marcus Kaiser's lab. I don't know if you know Marcus. And by the way, Marcus will also be one of the invited speakers tomorrow. So we'll have a continuation in terms of topics. And one of the things that struck me as really interesting, by the way, I'm a computer scientist by background. So I was working more on the algorithmic aspects of finding network motifs as possible patterns that could characterize brain networks. But nevertheless, when we were talking about structural connectivity, I was really intrigued by the fact that at that point in time, Marcus told me that it could have some implications on actually surgeries, physical surgeries that could actually alter the topology of the brain. In that case, it was more epilepsy, right, which was relatively, like, say, in a very simplified fashion, like more dense networks, we could actually cut parts of the brain, which is kind of scary, I guess, right? But I would guess I would like your opinion on whether some of these discoveries may actually lead to treatments that are not just necessarily drug related, but for instance, actually physically existing.
That is a very good point. And I would say that people are still kind of scared to think of this cutting of connections in the brain, because basically that's what Egas Muniz, you know, the Portuguese who won the Nobel Prize, he found that if you cut, like really here in the brain, you cut the connections from the basal ganglia to the frontal cortex, you can reduce the psychotic behaviors. Of course, the people were becoming apathic. But what was wrong at the time is that this was done by, you know, trial and error experiments without knowing why this is happening, you know. And what my intuition is that the more we understand, and if we know exactly where to cut, where to touch, where to modulate, I hope we can find non-invasive techniques, right? These ideas of transcranial magnetic stimulation. But so far it is also still done a bit, I mean, without knowing exactly. So it's hard to predict the outcome, because so you apply a stimulus here, but you know that if you apply in different places at different frequencies, the outcome is different. And then to do studies, the clinical studies, it's very hard because you cannot try all the possible stimulation techniques and see what comes out, right? Like cutting, you cannot cut everything in living humans and see how it comes out, what comes out. So if we have better models, if we understand better what is going on, how the network dynamics, you can know exactly where to cut, where to do a small incision that may solve, right, the wrong network loop, for example, that is being engaged and that is giving tremors, or yeah, there are different or anxieties. I don't know, hopefully yes.
Thank you.
Thank you.
Hi, Joana. Thank you for the talk. As someone who's network science, who's looking at network neuroscience, how would you suggest the best way to approach it? Because I've looked into it, and there's like a variety of pipelines about how you construct these, and a variety of approaches how you analyze them. And being someone who isn't a neuroscientist by background, but has like a kind of mathematical background, who wants to go into it, what is the best way to approach it?
So this is also a very good question. To be honest, so when I started to be, so the first paper I was also not directly neuroscience background. I'm a biomedical engineer, and we were given just the correlation matrix of the signals, and we were given the structural connectivity matrix. So just how the different brain areas connect to each other. So I would say, of course, it's a very simplistic thing. Then for me, it was more and more important to look at the signals and to see how the signals are behaving. but I would suggest using like a parcellation of the brain that divides the brain, for example, in 100, 200 nodes. There are different ones. One, the most widely used is called AAL, Automated Anatomical Labeling. It divides the brain into 130 something areas, depends. There is now a new version of it. And knowing how the structural connections, so there are probably already open source data sets on that, I may suggest, or if you establish a collaboration or ask someone. So to know the network structure, right? How the different brain areas are connected together, using also the centers of gravity because the positions may be important, the distance between areas as well. And then you can simulate using, you can directly analyze like we were saying before, you can directly analyze that network, just applying, which is already complex, measures from graph theory. But to be honest, I think that has been done already. Like there were really thousands of papers, I would say, applying graph theory to different types of measures. So seeing, for example, single subject variability between subjects, how this changes between groups. Then looking at the signals in those brain areas, how they interact together. And there's functional MRI. There are many issues now, at least from my side. What is the right pre-processing technique to apply to the data? So now at this stage, I'm like, no, I want raw signals. I'll do then my own pre-processing. But of course, it's a bit bad. But to know what to model, you need to understand what are the different steps. Of course, then I want to understand how the different steps were done and their implications. But yeah, I would suggest just taking a person and looking at the signals from the 100 areas and really seeing that sometimes the signals in some areas go up, in the others go down. So just correlation. So there has been also a hot topic of debate is like why people assume that correlation is functional connectivity. Like if we're talking about multivariate complex systems, we don't need in-phase correlation, you know, without any lag. So also these phase relationships, delayed interactions. But there is space in the field to grow and to develop new models and new techniques, like, because there has been developed dynamic causal modeling and this, but it lacks, it still has limitations. The gold standard methods haven't arrived. So I think it's a bit a very free field of research because you are able to, as long as you provide evidence that it returns a good distinction between disorders or between conditions, or yeah, try to use an open source data set. That would be my suggestion, that has structural connectivity and functional signals.
Thank you.
You're welcome.
Okay, I think we have a very interesting round of questions. So
yes,
thank you again, Joana, for your wonderful
Thank you
talk.
so much as well for inviting me. It was a pleasure.
Maybe we want to thank the speakers just once again.
Thank you.
Okay, and Joana, I will also, if you give me your PDF, I'll make it available to all the audience so that they can actually reach you after this. Okay, but I will contact you.
The PDF of the presentation.
I'll
Yes,
let
okay.
you know so that anyone interested can continue working. Okay, so thank you again so much, and I hope you also be able to enjoy the rest of the conference and even being online. Okay.
Yes, I will. Thank you. Thank you.
So in our case, this is the day where we have the earliest coffee break. So ideally, this next coffee break would be splitting into halves the next talk. So we didn't want to do this to Fernando, and we didn't also want you to have a coffee break before the last talk, or else some of you might run away. So we want then to also have audience. So I apologize for being a bit early coffee break, but I guess it's a good thing, so you can have more coffee after lunch and not sleep, hopefully. Okay, so we'll continue at 4. Okay, so feel free to rest over here. I will go and check if the coffee break is ready because we are a bit early, which is a good thing. So I hope you are enjoying, and see you in a bit at 4.